Thank you, Daniela. Let's see if we can get. Oh, here we go. Good. So it's a pleasure to introduce Dr. Mike Schweizer, who's an assistant professor in the Department of Medicine. He's a medical oncologist at the University of Washington, and he's going to describe some really fascinating work of using blood-based markers, circulating tumor cells, and cell-free DNA to give insights about tumor biology. Mike?
All right, thanks everyone for coming out on a rainy Saturday. What I wanted to talk about was liquid tumor biopsies, which is basically using information about cancer that we can glean from a blood draw to help inform precision medicine. How can we use blood draws to help allow us to tailor our treatment approaches for an individual patient, sort of moving away from that one-size-fits-all model. And so what is precision medicine? So I just copied this from the NIH website, but they define it as an emerging approach for disease treatment and prevention that takes into account individual variability in genes, environment, and lifestyle for each person. And that's really the key, is that this is for an individual. It's not for, picking a drug that's going to be good for everyone. It's about picking the drug that's right for an individual. And so when we talk about precision medicine in the context of treating prostate cancer, really the majority of strategies rely on dissecting the molecular events, understanding the biology of what drives an individual cancer cell. And that usually means evaluating changes in tumor proteins, RNA, and DNA, allowing us to differentiate what's going on in the cancer cell from the normal cells in your body. And this allows us to understand what drives drug resistance, which is really essential to developing better therapies, and also allows us to assess for vulnerabilities that might be present in a cancer cell, but maybe not in a normal cell. In theory, it allows us to identify so-called Achilles heels that we can target. And so, just to make sure we're all on the same page, when we think about studying a cell, and in this case a cancer cell, there are really three key molecules that I think we pay the most attention to that really give us a pretty good picture as to what's going on with that cell and why it functions the way it does. The DNA is sort of analogous to the blueprints, okay? The RNA is basically a way to take those blueprints and translate them into the final product, which would be, in this case, protein. I have a house here for this analogy, which is probably not really that accurate. Proteins are a lot more dynamic than that. They're functional particles. They basically are the workhorse of the cell. It probably would be better to have a machine up here, but then I couldn't use a construction worker and the whole thing fell apart. So I kept it as is. So one of the major challenges, though, is that to study these cancer cells, we've traditionally relied on doing metastatic biopsies, actually going in there and doing a surgery to remove a tumor or sticking a needle into a tumor. And that poses a lot of challenges. Biopsies are costly, they're very time-consuming, and I think more importantly, they can be painful, and there's potential other complications that can arise from doing those types of things. And so I think we need a better way to really assess an individual's prostate cancer, ideally one that can be done repeatedly and in real time. And so I think the real solution to that issue would be to focus on liquid tumor biopsies, so obtaining tumor material, usually DNA or actual cells, directly from the bloodstream. And so with liquid tumor biopsies, basically what you seek to do is evaluate cancer biology based on information that's in the bloodstream. So if you imagine there's a tumor that sits just adjacent to a blood vessel here, this tumor is actually constantly shedding cells and also DNA and other particles as well into the bloodstream. And when you draw the blood from a patient with cancer, you can actually collect some of these tumor cells and you can collect DNA and you can study them. And so in some ways this allows you to circumvent the need to actually stick a needle into the tumor itself, and you can get a lot of information from a blood draw, which is, relatively speaking, a lot simpler than doing a biopsy. And so again, with circulating tumor DNA, or ctDNA, this is really looking at tumor DNA that's present in the bloodstream. Now this is always mixed with normal cell DNA, and the normal DNA is always in a lot higher abundance than the tumor DNA. But we are developing technology that allows us to actually isolate out the tumor DNA and identify it as such so that we can really get insights as to what's going on in the cancer as opposed to what's going in 99% of the DNA in the circulation, which is your normal cells. One advantage of this is that it is detectable in most prostate cancer patients. It can be challenging to actually get useful information from it in somebody who doesn't have high PSA or more cancer on scans. But for the most part, you can at least detect it on some level. One of the downsides is that it only provides information about DNA. You're not learning anything about proteins in the cancer cell. You're not learning anything about RNA. So it's a little bit of an incomplete story. Circulating tumor cells, on the other hand, are whole tumor cells in circulation. These are relatively rare. They've been estimated to be about 1 in tens of millions of normal cells are the circulating tumor cells. These are not detectable in all prostate cancer patients. You generally only can isolate them out from patients that have more advanced cancers that you can see on scans and that have higher PSAs. But ideally, these cells are actually ideal in some ways because they provide a more complete snapshot of what's going on in the cancer. You get information about DNA, RNA, and also protein. And so, one of the challenges really with studying circulating tumor cells is isolating them. And so there are actually a lot of platforms out there that seek to allow us to collect these tumor cells from the bloodstream. In general, I would say that most rely on distinguishing tumor cells from normal cells based on the proteins that are on the cancer cell's surface. And they do this probably in most cases using antibodies, which are specific for cancer cell proteins. And so we've focused on a couple of these technologies, and I'll walk you through 2 of them that we've been doing some work with. The first circulating tumor cell platform is called RareSite. Basically what you do is you load blood into a centrifuge tube, you spin it down, and then what you're left with is basically in the bottom of the tube you've got all the red cells, and then at the top of the tube you have all the nucleated cells, the white cells and the cancer cells. And so you take this little vial of the tumor cells mixed with white cells, and you actually spread this across a microscope slide, and then you stain this slide with a cocktail of antibodies that allow you to discriminate what would be a normal cell versus a cancer cell. And then you actually load this into an automated microscope, which uses a computer basically to scan the entire slide, and it uses software that's pretty similar to facial recognition software to identify what it thinks is a cancer cell. And so this system will then present the user, the person that's looking under the microscope, with cells that it thinks are cancer cells. And if the user agrees that, yeah, it looks like a cancer cell, they can actually pluck that cell. So right here, the computer said, this looks like a cancer cell to me. The person who's looking through the microscope says, yes, I agree. And then it's got a little mechanism for cutting this out of the slide and retrieving it, and it can plop it into another vial so that you can actually do some additional work, like studying the DNA in that cell or studying proteins and things like that. The next platform that we've been using is called the ADNA test, and this is a platform that is really ideally suited for studying RNA in cells. And so the way that this one works is you take your blood and you load it into a tube, and this green circle here would represent 1 cancer cell in the vial. You load it full of antibodies that are attached to magnetic beads, and so these antibodies are specific for tumor cells, so they only bind to the tumor cells. And now that the tumor cell is coated in these magnetic beads, basically, you can use a magnetic source on the outside of the vial to basically isolate out the tumor cell, discard all the cells that aren't attached to the magnetic beads, and then you basically have a purified sample of just tumor cells in the vial. At this point, you can lyse the cell, you can pop it open, collect the RNA, and then you can do additional studies to learn about what's going on with the RNA in that cell. And so this sort of background. I wanted to walk you through maybe a couple examples of how we're using this technology to try to inform precision oncology clinical trials that we have here. Really, the goal is to develop personalized strategies for treating prostate cancer patients. And again, I think that liquid tumor biopsies can be really helpful in this regard because, again, you don't need to stick a needle in somebody's tumor every time to get this information that's going to allow you to learn about what's going on with the cancer. And so the first example is using platinum-based chemotherapy for patients that have what we call DNA repair deficiencies. So DNA repair is basically an essential cellular function. If your cell wasn't able to repair DNA damage, they wouldn't survive beyond a couple of weeks probably. We wouldn't live into our 80s on average. So the reason is that throughout a person's life, DNA is constantly damaged. And this can happen because cells are dividing and that induces some stress. It can also happen because you're exposed to carcinogens like ultraviolet light, or if you're a smoker, tobacco smoke, and there's other things too. And so when that DNA is damaged, the cell needs to be able to repair it, otherwise it's going to die. And so what we've learned is that a number of cells, and probably more so with cancer cells, they have impaired ability to repair DNA, and if they have specific mutations in genes that are essential to this DNA repair process, that may represent a major vulnerability that we can exploit. Some of these genes that are essential to DNA repair are things like BRCA1, BRCA2. They're also referred to as BRCA1 and BRCA2, and you may have heard them in the context of being an increased risk factor for developing breast and ovarian cancers, but we now know that they're also very important to prostate cancer biology too. And so the idea would be that if you treat a person with platinum-based chemotherapy or another DNA-damaging agent, that agent's going to induce damage to the DNA, and in one scenario, if a cell has intact DNA repair machinery, so it's able to repair the damage, some of those cells are going to die, but in general, the scales are tipped towards many of those cancer cells surviving, actually. So the therapy is not actually all that effective. Now, conversely, if you take a tumor cell, a prostate cancer cell that you know has a mutated BRCA1 or BRCA2 mutation that has impaired DNA repair machinery, and you subject it to a DNA-damaging agent like platinum therapy, now the scales are tipped towards more of those cancer cells dying, and we know that this type of therapy is much more effective in these patients. And so this is just an example that was published by Dr. Heather Cheng that sort of highlights this. So this is a guy with advanced prostate cancer who has received several of the approved agents for prostate cancer, abiraterone, enzalutamide, and docetaxel. You can see that basically over the ensuing 2 years, he's had a progressively rising PSA this entire time. And so around this point, they decided to do a biopsy, okay, not a liquid biopsy, just a conventional one. But they discovered that he had a mutation in one of these DNA repair genes, this BRCA2 gene. And so based on this, they predicted that he'd probably respond very well to platinum-type chemotherapy. And lo and behold, when he's treated, his PSA declines rather precipitously, and he's done quite well with this type of treatment. And so this type of therapy doesn't work for probably most prostate cancer patients, but if you have this type of mutation, we think it works very well for them. And so Dr. Cheng has actually developed a clinical trial to try to sort of prove this further. And basically the way this is set up is if a patient has one of these DNA repair deficiencies, they're allowed to enroll to the study in which they get a platinum-based chemotherapy. And again, we think that that's going to predict for them having a good response. And two important points about this study is that it does allow guys to enroll if they have one of these mutations detected from ctDNA, so DNA in the bloodstream. And then another important point is that everybody on the study is going to have blood collected at the beginning and at the end of the study so that we can look at their circulating tumor cells, their ctDNA, and really try to understand the biology even better and maybe develop the next iteration of this that's going to be more effective. The next example is some work that I've been doing looking at targeting androgen receptor splice variants. And so just as some background, if you imagine this is a normal prostate cancer cell, this is sort of a simplified way to think about its general biology. So the prostate cancer cell is here. The main driver of its growth is the androgen receptor, and in order for this to work, you have to have androgens, the one that most people have heard of as testosterone, bind with this receptor. And so these androgens or testosterone in circulation migrate in the cell, then this complex goes to the DNA, and that basically sends off signals saying that you should grow and cause problems, okay? And if you have a guy who's on hormone therapy, basically they all work by preventing this process, right? So drugs like Lupron are going to lower the amount of androgens in circulation, so it's not there to even bind in the first place. Drugs like Casodex or enzalutamide actually physically block this interaction so that even if testosterone gets in the cell, it can't hook up and drive the cell to grow. And so that's how those drugs work. But the problem is resistance develops to pretty much all hormonal therapies. And one of the mechanisms that we know as being relatively important is the emergence of these androgen receptor splice variants. Essentially, the way this works is that if you imagine a normal androgen receptor has this nice groove in it where testosterone can bind, these splice variants don't have the groove at all. And so testosterone has nowhere to bind here. And the important thing to understand about splice variants is that they don't care that they don't have the groove to connect with testosterone. They're still going to drive the tumor's growth. So essentially, every drug we have that works by blocking this interaction is not going to be effective because there's nothing to even block in this case. And so the idea is you have a cancer cell, it's got a splice variant now, testosterone goes in the cell, says, I don't have anything to bind to, the splice variant doesn't care, it still goes to the nucleus and tells the cancer cell to progress. And so hormone therapies are thought to be ineffective in this setting. And this is just some work that was published in 2014 out of the group from Johns Hopkins looking at the presence of androgen receptor splice variants as detected using the ADNA test, that assay I told you about earlier. And basically what they showed is that whether you're on enzalutamide or abiraterone, you basically are not going to have a good response if you have the presence of one of these splice variants. These graphs are basically, each bar represents a patient. If it's going down, that means the PSA is declining. If it's going up, it means the PSA is rising, which is bad. And in this case, the blue bars are guys that don't have splice variants, and the gold ones are guys that do have splice variants. And you can basically see that most of the gold bars are going upward, meaning that even though they got abiraterone, the PSA is still rising. The drugs don't work, basically. And so because of this, a lot of people have been interested in trying to develop therapies that can be effective for guys that have splice variants. Is there a way that we can actually target the splice variants themselves? And so this was one study where they actually took 1,100 FDA-approved drugs, and they basically dropped them onto prostate cancer cells that had these splice variants and looked to see if any of those drugs could inhibit those cells. And so they identified actually one drug that worked pretty well at doing this, niclosamide. This is actually a really old tapeworm medication, okay? But it looked like it actually was good at inhibiting androgen receptor splice variants, so they did some additional work, and they looked, again, in splice variant positive cell lines, and they showed that when you treat those cells with enzalutamide, they don't respond, which is to be expected because they have the splice variant. But when you add niclosamide, all of a sudden, those cells are dying. And so the idea is that combining niclosamide with enzalutamide might be a good strategy. And so I designed a trial to look at this a little bit closer, and this is an early phase study that's looking at using high-dose niclosamide in combination with enzalutamide for guys that have already progressed on abiraterone. So these are folks that have really received some of the most modern treatment and are still having progressive disease. And the idea is, can we target one of the key resistance mechanisms in this setting? And so, importantly, in this study, we are also planning to collect circulating tumor cells at the beginning and at the end. And the idea is, can we look at what's going on with these androgen receptor splice variants? Are guys that having the splice variant still responding well to this treatment? And does niclosamide have some way to modulate the splice variant's expression? And so this allows us to sort of learn a little more about the biology of the cancer cell and the role that splice variants play, and importantly, to see whether or not this drug is able to effectively target them. And so the last example is really just to highlight that we are, continuing to work to understand the biology of prostate cancer, and I think liquid tumor biopsies is one of the key ways that we're going to be able to expand this to more patients. And so I'm running a study where we're looking at collecting blood draws for liquid tumor biopsies from a number of guys who are starting on either abiraterone, enzalutamide, or an experimental prostate cancer drug. And this is a complex figure, but the key point is we're basically collecting blood at baseline before they start treatment, about 12 weeks in when we think they probably are going to be responding well, and again after the drugs aren't working anymore. And really this highlights, I think, the power of liquid biopsies is that you can collect blood samples serially for an individual and really get an idea as to how their cancer evolves over time, how resistance emerges, and this will hopefully allow us to develop more effective strategies to treat patients moving forward. And so just to summarize, I think precision medicine, I think, is really sort of where oncology is heading as a field. The idea is that we're moving away from this one-size-fits-all and actually developing a therapy that's specific for an individual. And I think that metastatic biopsies are still important, but they're challenging, and it's not going to be the right thing to do for a lot of patients. So hopefully liquid tumor biopsies will dovetail nicely with that and allow us to forego biopsies in at least a number of patients. And again, there's a lot of ongoing efforts to try to sort of leverage this approach. I've highlighted a few here, but there are many more that we're working on. So that's all I got.
Wonderful. So if we could have the speakers from this morning's second session come up, write your questions down.